When it comes to looking at microscopic life, there are two ways you can go. The first is to bring in a sample of your local pond water and then search thoroughly through it looking for something that's alive. When you finally find something that's living, you now have the problem of trying to identify what you see. A much easier way is to order living specimens from a science supply house. They're not very expensive, $2 or $3 for a little container like this one that's full of amoebas. There's plenty here for you to use and other teachers in your school. They'll last for 1 to 2 weeks, and you know what you're looking at. You know they're amoebas. It's just a matter of getting them from there onto the slide. Now, some of the other considerations to take into account when you're choosing which microorganisms to look at, especially if you're using a microprojector. You want to have living things that are fairly large. Amoebas are good examples. You also want to have living things that don't move around too fast, so you're not constantly chasing them with the mechanical stage, trying to keep them in the field of view. The ones that I have chosen are ones that I think you'll find very useful. It's the Amoeba, the Stentor, the Paramecium, the Sporostomum, and the Volvox. These are all very interesting microorganisms. Now there's different ways to prepare slides, and we should probably cover that too. If you have your box of flat slides and a little box of cover slips, incidentally these are 18 by 18 millimeter and they're #1 cover slips. The first thing you do is take your sample and put 1 drop on your slide. Then take the cover slip just like we did earlier in this tape and carefully try to keep your fingerprints off of it. Carefully lay it down and let it drop at an angle. And that should make a nice smooth flat surface so you don't have to do a lot of focusing. It kind of compresses the water out, and that's one of the main reasons for a cover slip. If you have a well slide which looks like this, you see there's a little indentation here. You can just put the drop in there. Many of these larger living things will get squished if you put a cover slip on them. So you either have to use a well slide alone or just a drop of water on a plain slide with no cover slip. Other times we want to trap some of these living things that are moving a little bit too fast, like the Paramecium, kind of put it in a little corral. So in this case, we'll take our drop of sample, we'll put it on the slide, and then we'll take a tiny piece of cotton from a cotton ball and carefully, just a few strands mind you, lay it along the liquid in the sample and then put the cover slip over the piece of cotton like this. We now have a lot of little barriers to keep the Paramecium trapped in different spots, and you'll see when we look at the Paramecium how useful this cotton can be. A third possible thing you might try doing, particularly with Paramecium that are moving quite fast, is try a chemical called methyl cellulose. You add 1 drop along with the sample before you put the cover slip on. You don't need the cotton, and after about 30 seconds or a minute it'll start slowing down the Paramecium. It affects them, and pretty soon they'll come to a standstill and you can look very closely at them. Now let's take a look at some of these samples. We'll start by looking at the amoeba. Amoebas usually live on the bottom or along the sides of the container, and they look like tiny little white specks. So we'll suck up a sample of water and then we'll look very closely and drop a few drops out, and maybe we'll see 1 little white speck in there. If not, we'll just have to keep trying different drops of water until we get one. I'm not going to use a cover slip on the amoeba because they're so large. We'll put it on the stage of the microprojector, and I'll set my turret down here to A, and I'll be using the smallest objective lens, the 16mm lens. We'll use our low power lens first to find the amoebas, and they look like tiny little gray blobs. Once we've located them, we move up to the high power lens so we can take a better look. The amoeba, once again, is a single-celled organism, and it has a jelly-like material inside the cell. This is called cytoplasm. The amoeba moves in a peculiar way. It extends a foot outward and then brings the rest of its body into the foot. The clear circular bubble is a water bubble. This is how the amoeba gets rid of extra water. When the amoeba grows to a large size, first the nucleus splits to make 2 nuclei, and then the amoeba itself splits into 2 amoebas. The next specimen we're going to take a look at is the Stentor. And once again, because of its size, we're not going to use a cover slip. As before, we start at low power to locate these single-celled microorganisms. The edge of the water drop is shown in the top of the picture. We will now go to a higher power and look more closely at this protist. The Stentor will either swim freely in the water or attach its narrow end to some debris. It has hundreds of tiny hairs on the wide end which it uses to gather food and move around. These are called cilia and form a circle around the mouth end. The Stentor is transparent, and at this magnification it is about 6 inches or 15 centimeters long. This translates to a magnification of about 200 times. You may notice other strange things in your drop of water. Because of their size, it may be necessary for your students to come up in small groups for a better view. The next specimen we're going to look at is the Paramecium. Like the Stentor, the Paramecium has those tiny little hairs called cilia that allows it to move around and gather food. But unlike the Stentor, the Paramecium is much smaller and moves around quite rapidly. So in this case, we're going to want to use the cotton and the cover slip, or we'll try the methyl cellulose. The final specimen we're going to look at is one of the most interesting because it's neither plant nor animal. It has traits of both. It's called the Volvox. The Volvox is a colony of many single-celled organisms connected together by fibers. Each cell has 2 whip-like flagella which beat in unison to allow the colony to move. The colony is spherical in shape and hollow on the inside. It not only moves like an animal, but it can also make food with chlorophyll like plants do. The Volvox is usually found swimming near the surface where there is more light. The large green spheres are new colonies that are developing within the parent colony. Eventually, the parent colony will die or rupture, and the new colonies will be released. At this power, the Volvox is about 8 centimeters or 3 inches across. Now that you're having lots of fun looking at tiny living things with your microprojector, it's time to leave the protist behind and move up to the plants and animals. One important aspect that your students should understand is that most living things are made of many cells, and the cells form particular structures, different parts that have a specific function. For example, you might have them take a look at an insect wing under the microprojector. When you start to look at insect parts, you're going to be opening up a whole new world of fun things to look at. To wrap up your investigation of cells, you may want to take a look at a Daphnia. It's a tiny little multicellular animal that's very interesting because it's transparent. The study of fossils is very important in that it allows us to indirectly learn about life on Earth millions, even billions of years ago. By studying fossils, we find out that the first life forms were the single-celled plants and animals. As time went on, new, more complex life forms evolved, and most of the ancient life forms, like the dinosaurs, ceased to exist. This may have been caused by adverse environmental conditions. Some of the tougher, more adaptable species continue to survive. The cockroach, for example, has been around for 350 million years. That's 349 million years longer than we have. Let's introduce fossils with an activity that explains how we learn things by observing indirect evidence. We'll call this activity Who Was There? Set up about 10 stations throughout your room. At each station, place various objects. Typical examples might be a bird nest or chicken egg at station 1, some pine needles at station 2, and perhaps even a man's tie and a newspaper at station 3. Student groups go around, make observations of the items that they see, and record the information. Try and make as many inferences as possible. Then when they're back in their seats, you can have a lively discussion about what type of person or animal or plant might have been there. The important lesson here is that we can learn a lot about living forms by studying the remains that are left behind. A fossil is the record or evidence of a plant or animal that lived in the past. Scientists determine the age of fossils by measuring the amounts of certain chemicals found in the fossil or in the rock surrounding the fossil. It may be a whole animal that was frozen in ice or fell in a tar pit. It could be a bone or a tooth that was hardened by minerals, or an insect that got trapped in the sap of a tree. A fossil can also be a print, like a footprint or the outline of a leaf. Your students will become more familiar with fossils if you let them make one of their very own. It's inexpensive, it's easy, and it's lots of fun. Here's how we do it. Start with a piece of flat clay on a plate. Give the student an object to impress into the top of the clay. Seashells work really well, especially the scallop type, because they have all the ribbing on them. If you can't find seashells, you might try walnuts or bones. Once the object has been pressed into the clay, you remove the object and place a paper cuff around the outside of the clay. Press down on the paper cuff so that it goes into the clay. We don't want any of the plaster rolling out from underneath the cuff. Then you're ready to mix the plaster. Mix 2 parts of plaster to 1 part of water. You want it to have the consistency of pancake batter. Incidentally, about 5 pounds of plaster is good for about 50 or 60 different casts at this size. You pour the plaster into the top, wait for about 30 minutes till it's hard, and then pull it off. You'll end up with something like this. You have a very nice cast of your object. Another technique is to make a mold instead of a cast. This time we'll take a very small paper drinking cup, pour our plaster into it, fill it about half full, and then carefully drop our seashell into the top. Let it float on the top. After it hardens in about 30 minutes, we tear the cup open and we have made a very nice mold like you see here. To keep the object from sticking, it may be necessary to first coat it with petroleum jelly or shortening. I would also recommend that if you're doing this in the classroom, you mix the plaster in the water to avoid the major mess. Your students may notice that when this reaction occurs, when the plaster solidifies, it gives off heat. It gets warmer. This is actually physical science. A chemical change is taking place and heat is being given off.